Hello, and welcome back to Scientific Ethics. What we're going to do today is switch gears a little bit. And so for the last probably 4, 5, 6 lectures, we've been talking about organ transplants and stem cell therapy as a way to kind of treat human disease, to treat damaged cells or damaged organs. Now we're going to change directions for kind of this next section and continue that on for the remainder of the course. So we're going to be looking at some of the more recent advances within DNA, genetic screening, genetic modifications. And so in order to be able to understand that, and again, recognize that this is kind of a non-majors course, we need to talk a little bit about the science, a little bit about what DNA is. And so don't worry if you're not a scientist. It's not going to be overly complicated. I'm going to summarize this and hopefully make it user-friendly and provide you with information that I think is going to be really important for you to understand what DNA is, how DNA works, so that we can look at the applications of DNA going forward. And so don't be scared off by the science. I promise you it's doable. It's definitely something that, at least from my perspective as a geeky biologist, I think is going to be interesting and potentially, hopefully, interesting to you as well. So we're going to start out today taking a look at basic concepts of DNA. And the first part of that is to be able to understand the basic structure of the DNA molecule. Now, if we take a look at this in a very broad sense, what we're going to see is 3 types of biological molecules that are going to be incredibly important for what the cell is going to be doing. And what it's going to be doing is controlling the activity of the cell. And so we're going to have DNA up here at the top. Our DNA is going to be fancy scientific name, deoxyribonucleic acid. This kind of describes what the molecule looks like. But don't worry about that. Just think about it as DNA. The DNA is going to be found within your cells, within a specialized region of the cell called the nucleus, which is going to be important for carrying that information and storing that information, making sure that information is available to you throughout your life. It's going to be kind of a variation of the DNA, essentially half of this DNA that we're going to be transmitting to our offspring, either in our egg or our sperm. And that's going to kind of dictate what characteristics our offsprings are going to have. So you can think about our DNA as kind of being like the encyclopedia of all the information that's needed by the cell. RNA is the second molecule down here. RNA, again, fancy scientific name, ribonucleic acids. You can see it's similar to this. You can see they're both acids. You got some nucleic, ribonucleic. Don't worry about that. Just kind of describes it. You know what is DNA and RNA? So DNA is like the encyclopedia. RNA is like the working copy of the information that we're working with. And now with the internet, this analogy kind of isn't a great example anymore because you can get whatever you need on the internet now. Don't use Wikipedia, even though Wikimedia has a lot of images that are free that we can use in these diagrams. Think about DNA as being the encyclopedia. And when I was going through college, if you wanted to look something up, we didn't have the internet available to us like you have it available now. We would go look it up in an encyclopedia, look it up in a reference source, and you couldn't check out the encyclopedia. So the DNA is like the encyclopedia. It's going to be in the nucleus like the encyclopedia is within the library on campus. But when we want to use the information, we can't check out our encyclopedia. You can't check out the DNA because we might lose it, and that would be bad for the cell. So what we do is we make a temporary copy. We're going to copy this information that we want from the DNA into the RNA molecule. And we're going to use this RNA molecule, this temporary copy, to do what it is we need it to do. And so if I needed information, I would go to the library, check out find the encyclopedia, find the section I want, and then, if I want to study aardvarks, I go to, the encyclopedia that covers A, go to the page on aardvarks, and then I would, we still had copy machines, they're old and slow and all that, but I would copy the information, the pages associated with aardvarks, and then I could take them back to my residence hall room or take them to another location and refer to them as I was writing my paper down here or doing something like that. The same type of thing occurs within the cell. We're going to have the DNA stored within the nucleus. That's going to be the information that's important that we make sure we don't lose it. So we're going to keep it there. If the cell needs it, we're going to go in and make a temporary copy of the section we need, and that's going to be in the form of RNA. And then we're going to take it, as a blueprint or as the information we need to another location of the cell. We're going to use that information to build the proteins. The proteins are going to control what the cell is going to be doing. And so we can see that the DNA is going to be incredibly important for what we're going to be doing if we study what's going on with these proteins. We got to fix these proteins or kind of change the expression of these proteins to treat a disease or treat a condition. So DNA then is going to be very, very important as our encyclopedia of information for our cells. Now, if we take a look at this, again, fancy scientific word up here, nucleotides are going to be the basic subunits of the DNA molecule. And so basically what this means then is that they're going to be like chemical building blocks. And so they're almost like little Legos that we're going to click together, or building blocks that we're going to click together in a long chain. And we're going to have 4 different nucleotides. We're going to have adenine, we're going to have guanine, cytosine, cytosine over here, and thymine over here. And this is going to be a portion of the molecule that we're looking at. And these are going to be what's different between one subunit, one of the different nucleotides and a different nucleotide. Again, looking at these as the subunits. So there's more than molecule that's going to be present, but we can take a look at it. And again, without looking at all the different details, we got kind of an NH down here. Don't worry about it, but it's just, a different shape here. We've got an O here, that's a different shape, but the rest of it kind of looks pretty much the same, a little different kind of over here at this point. Underneath the region here, we've got like this single got 6 sides, so it'd be a hexagon. We've got a 6-sided structure over here that's a hexagon. But again, this one, we've got the NHH group, 2 H's and an N up here. We've got an O here, we've got other changes. So these are kind of the important portion of our nucleotide molecules. And just recognize they're going to be a little bit different, whether we're talking about an A, a G, a C, or a T. So I'm not even worried that the adenine, thymine, guanine, or cytosine. Recognize it as the A's, the T's, the G's, and the C's. And so these are going to be the important subunits that we're going to hook together to make our DNA molecule. Now, if we take a look at this, our DNA molecule is going to be double-stranded. And so we can take a look at it. We can see our A's over here, our T's over here. We can get a C down here at the bottom and a G down here at the point. And so basically, we've got these kind of strands that are going to be present of nucleotides over here, and another strand of complementary nucleotides over here. They're going to give us this almost like ladder type structure. And so this backbone of portions of the molecule that we didn't talk about are going to be hooked together, forming like the vertical sides of a ladder. And then those A's, the T's, and the G's, and the C's are going to be kind of like the rungs of the ladder. They're going to interact with one another. They're going to hold these 2 strands of DNA, these 2 kind of vertical sides of our ladder together. And they're going to be complementary. So where we have an A on one strand, we're going to have a T. So if we got a T down here, we're going to have an A over there. And where we have a C, we're going to bind to a G. If we have a C here, we're going to bind to another G over here. And so we've got some rules on how this molecule is going to be working. But the important thing, again, all of this backbone is kind of structural. It's going to be generic and it's going to look the same regardless of which portion of the DNA molecule we're looking at. The important things are going to be the A's, the T's, the G's, and the C's, because this is going to be where the information is going to be coded or kind of hidden within the nucleotide molecule that we need to recognize within the DNA molecule. Now, what's going to happen, if I go to the previous slide, we've got this nice flat ladder here, making a nice, kind of artist rendition. Reality isn't always a nice artist rendition. And so we've got this ladder that we can look at it as kind of flat. Again, the rungs of the ladder, the rungs kind of here, the A's, the T's, the G's, and C's, the parts that you stand on to get the ladder. We got the vertical sides of the ladder kind of over here. When reality, what's going to happen is we're going to take this and we're going to twist it a little bit. And so the DNA molecule is going to be a double helix. A helix is like a twisted around itself molecule. And so what we're going to have is that ladder's going to be twisted. It's going to be kind of spun around like this. But along the inside, we've still got those binding sites where the A's bind to the T and the G's bind to the C. And that's going to be that important information to the inside of the ladder. So this is essentially just a ladder, but we've twisted it around as we're going. Now, we're going to have our DNA molecule, and we want to store it within the nucleus, similar to the way we want to store our encyclopedia within the library. And the way we're going to do it is we're going to take our DNA molecule, this long molecule here, that's going to be very long, very fragile, capable of being broken. And what we're going to do is we're going to wrap it around some specialized protein beads. And so these are going to be little beads that are going to be found within the nucleus that are going to allow us to wrap kind of the threads of the DNA around it so that they don't get broken and they don't get twisted and tangled and knotted. And so we're going to be able to store it. And so basically what's going to happen then is that if we look in the nucleus, what we're going to see is going to be referred to as a chromosome. A chromo for color, some for body. These are like colored structures that we see within the nucleus. We use a stain to look at biological molecules. And so like our encyclopedia, we're going to open up our encyclopedia to the pages we need. We're not going to pull every page out of the encyclopedia. If we look at our DNA molecule, we're only going to open up a small portion of it, and we're going to leave the rest of it kind of wrapped around these protein beads, again, because we don't want it to get broken, we don't want it to get lost. And so we have a mechanism in place that we can get to a very specific region of the DNA molecule and get to the information that's available, similar to going to the encyclopedia, turning to the page of aardvarks and finding the information on aardvarks that we're interested in. Now, when we're taking a look at the chromosomes, again, the kind of completely storage location of the DNA, so wrapped around it very tightly within these protein beads within our nucleus, we can take a look at what's referred to as karyotyping. And so you can basically look at the nucleus of an individual cells and study their chromosomes. And what we're going to see is that the human cell is going to have 23 pairs of chromosomes. And so we're going to have, and these are numbered from 1 to 22, and then we're going to have sex chromosomes here, the X and the Y. But what this means is we're going to have 22 what are referred to as autosomes. And the autosomes are basically that we have 2 copies of each. We're going to have 1 copy from our mother and 1 copy from our father of chromosome 1. We're going to have 1 copy of chromosome 2 and another copy of chromosome 2. 1 is coming from the mother, 1 is coming from the father. And we don't know which one it is unless we go through and study the genetics of you and your parents. But we know that under normal circumstances, everybody is going to have this kind of matched up set of their chromosomes. So you've got 22 sets of autosomes, and then we've got these sex chromosomes over here. And this is going to be a little bit different. We've got an X chromosome, which is relatively long. We're going to have a Y chromosome that's relatively short. And so a female is going to have 2 X chromosomes. So they have 2 of these long kind of squiggly things, where a male is going to have 1 X and 1 Y. And so if we take a look at this karyotype, we're going to see the 22 autosomes all paired up nice and kind of lined up on a chart like this. And it doesn't look like this in the cell. This is kind of taking a picture of it and cutting and pasting it and kind of lining it up like this. But we take a look at this, we've got an X and a Y chromosome. So this is going to tell us that we've got a karyotype of a human male. Now, one way that we can look at kind of genetic conditions is at a very broad sense. We can take a look at the chromosomes. And so what we've got with a patient with Down syndrome is instead of having just 2 copies of an individual autosome, 2 copies of chromosome 1, 2 copies of chromosome 2, again, 1 from the mother, 1 from the father. What happens in a patient with Down syndrome is that they actually have 3 copies of chromosome. They have 3 copies of chromosome 21. And so by taking a look at their karyotype, we can take a look at it and say, okay, we now have a situation where instead of 1, 2 of these chromosome 21s, we've got 3 copies of chromosome 21. So we have 3 of these DNA molecules. And because we have 3 copies of that, it's going to result in kind of excess expression of some proteins. We're going to see kind of changes in kind of genetic characteristics and the protein characteristics of this individual associated with having this extra copy of chromosome 21. And so that's an example of a genetic condition that can be identified by taking a look at the karyotype. Another one is Fragile X syndrome. And so you can take a look at that X chromosome. Normally, it kind of goes along squiggly, and we're not looking at Fragile X in this karyotype. But in some individuals with Fragile X, they've actually seen a portion of their chromosome break off. And if that portion of the chromosome breaks off, we've lost that information that that X chromosome normally carries. So that's like going to your encyclopedia, and I go, you don't do that anymore, but going to the encyclopedia in the library and want to study an aardvarks, and you find out that somebody's torn out those pages. And so if they've torn out those pages, we can't refer to that information. And if that's information that the cell needs, they're in trouble, especially if they're a male here where they've lost that information on the X chromosome and they don't have another X chromosome that would have a copy of it that's complete and kind of without the aardvark section torn out. So those are just two examples of chromosomal disorders that can result in kind of changes of the characteristics of the individuals. Okay, so that's kind of a background on kind of the basic structure of a DNA molecule. Come back for part 2, we're going to start to look at some of the genetic properties. So how can we get some information out of our molecule? As always, if you have any questions, feel free to email me. Thank you and have a great day.